Morning, everyone. So before we pick up where we left off last time, I just want to review some of the key concepts from B-cell immunity from the first lecture. So when we have antigen coming into the lymph node, it's coming in through the afferent lymphatics. And so this can be, if it's small, can be antibody complexes or antigen that's coated with IC3B. So anything smaller than a bacteria can come in through just passively diffuse through the lymphatics. The more important mechanism is when antigen is being carried to the lymphatics by macrophages and other cells. Okay, so they're going to enter from the afferent lymphatics. Now the first step is, if it's being carried there, is to pass the antigen off to subcapsular space macrophages. So here's the cortex, we have the subcapsular space, and those subcapsular space macrophages are the gatekeepers for transferring antigen to follicular dendritic cells. Now remember, if it's coming in as a small particulate that's smaller than a bacteria, it's going to go through the follicular reticular network and diffuse out through those holes in the pipeline. Now, the antigen is being transferred from the subcapsular space macrophages to follicular dendritic cells by non-specific B cells. And remember, at each step, the subsequent cells have higher levels of complement receptors and have different complement receptors, so they're essentially stealing the antigen from the previous cells. Okay, so that's how antigen gets into the B cell zones of the lymph node. For the B cells, they're going to come in through the high endothelial venules, and these are in the paracortex region, and they have additional migration signals that tell them to go to the B cell zones in the cortex. And this is where they initially get activated on follicular dendritic cells, and they're going to recognize both the antigen and the IC3B. So you have two different mechanisms for activating a B cell. The antigen-specific part, and then the complement part tells the B cell, yes, this is in fact something you should be making a response to. So when that B cell gets activated, it heads towards the T cell zone. Now, early in a response, if their T cell response hasn't been engaged, there's no T cell help. Or if this is a non-protein antigen, there's nothing to present to a T cell, so the T cells are not being activated. In this case, you get a T-independent B cell response, right? There's no CD40 signal from CD40 ligand on the T cells, and so you form in the paracortex these short-lived plasmablasts in the primary foci. These are making mostly low-affinity IgM, right? And what's the point of that? Well, it's to further start activating the complement so that you get more of this. Okay, so that you get more antigen being brought in by macrophages with complement receptors on them. So it's sort of the first step of the B cell response is really just to amplify the amount of antigen that's being brought in. Now, on the other hand, if it's later in the immune response and T cells have been engaged, or in a case where there is a protein and something to activate the T cells, which we'll talk about in the next couple of lectures, then there are T cells to provide that CD40 signal, okay? And so that CD40 signal to the B cell says, instead of forming the primary foci, sorry, I should have taken this off, that's not supposed to be there. Instead of forming the primary foci and plasmablasts, those cells go back to the germinal center. They start expressing an enzyme called activation-induced deaminase, which is responsible for somatic hypermutation and isotype switching. And then as they mutate those receptors, then they go and test them, and that's how you get affinity maturation. Now, the end result of a T-dependent B cell response in the follicle is the generation of long-lived plasma cells that make antibody for the rest of their lives, and long-lived memory B cells that sort of float around looking for antigen and make a response should you get infected with the same thing again. Okay, so are there any questions on that before we move on? Okay, so let's keep going. So when we look at a germinal center, these are specialized structures, okay? And the whole goal of a germinal center is to improve the antibody response so you get better antibodies, and then it allows for differentiation of the long-lived plasma cells and memory B cells. Now in the germinal center, we have the dark zone and we have the light zone, okay? The dark zone contains the rapidly dividing B cells, and we call those central blasts. Remember, whenever you use the term blast in immunology, it means it's rapidly dividing cells. And these are the cells that are mutating their B cell receptors. And this is how you're going to generate better antibody response. Now, as those cells move from the germinal center, or from the dark zone, they're going to move to the light zone. And this is where they test their B cell receptor to see if it improved the recognition of antigen. So this is the mutation area, and this is the testing area. Okay, and so in the testing area, those cells are not dividing. They and so we call them centrocytes. Okay, so centroblasts are rapidly dividing and mutating. Centrocytes in the light zone are the ones that are being tested. Now in the light zone, you also have follicular dendritic cells that are displaying the antigen and IC3B complexes on their surface. So that's where you can test if there's antigen recognition. But remember, what was the signal to go to the germinal center and start rapidly dividing? It was CD40. And so in the light zone, you also have that signal coming from CD4 T cells, which were only described in the past 10 years or so. We call those follicular helper cells. They're a special type of T cell that can provide that CD40 signal to tell that B cell go back to the dark zone and keep mutating. Okay, so now these germinal centers really only exist while there's antigen there, okay? So once the infection goes away and you eventually run out of antigen, then that germinal center disappears, okay? But you can find these in almost any lymph node because we're constantly making responses to our environment. Now, as I said, one of the key enzymes that's being expressed is a protein called activation-induced deaminase. And this has one job. It's going to take amine group of a cytidine base and convert that to a uracil. Okay, so that's the DNA damage here. Okay, and this is occurring in the dark zone. Okay, so in red here is activation-induced deaminase, and that's in the dark zone. You don't really see this out here in the light zone. Okay, so that's activation-induced deaminase is the protein that helps start somatic hypermutation. And then you turn this off when you're going out into the light zone to test if the receptor is better able to recognize antigen. So the centrocytes stop expressing AID, they stop proliferating, and they're testing, did that mutation make me better able to recognize antigen? And it does this by interacting with antigen on the follicular dendritic cell. Okay, so that's, can you see antigen? Now, part of this is that B cell has to have something that it can show to a T cell to get the next step, which is the CD40 signal, right? And so there's sort of a step process where it tests the ability to bind antigen, but then it's got to get more antigen so that it can process and present it on MHC class II to the T cells. So it gets an additional CD4 signal that tells it to go back to the dark zone. Now it gets this antigen from either follicular dendritic cells or subcapsular space macrophages. And it's doing this by trying to steal it. Now, this is where some of the confusion came about with these non-antigen specific B cells stealing antigen from the subcapsular space macrophages and transferring it to follicular dendritic cells. So it's not clear if the the antigen that the B cells are getting, that they're going to eventually present to T cells, comes directly from the follicular dendritic cells or from the subcapsular space macrophages. Now, essentially, this process keeps going on until you run out of antigen. Okay, so when there's no more antigen on follicular dendritic cells and the subcapsular space macrophages don't have antigen, then that's essentially saying, well, you've controlled the infection, so now it's time to disassemble the germinal center and go on with our lives. At that point, you have B cells exiting from the germinal center, and these become either long-lived plasma cells that are the antibody secreting cells, or memory B cells that are going to respond the next time you get infected with the same pathogen. Okay, so let's review somatic hypermutation. Okay, this key enzyme, AID, converts cytidine, to uracil, or in this case, it's uridine, in the openly transcribed DNA regions. Okay, and now part of the reason that these complementary determined regions or hypervariable regions are preferentially targeted, because these are open and accessible DNA structures. Okay, so AID can actually get in there. The other reason are the mutations show up there because those are affecting antigen binding, whereas the rest of it is just mostly structural, so it's not likely to make a better antibody response.
Sorry, Dr. Blackman, I have a
Yes.
question. For the previous slide about the dark and light zone, how these two zones divide or connect to each other?
So you can sort of think it as these cells are rapidly dividing.
Mm-hmm.
As they rapidly divide, they're pushed out of the dark zone into the light zone. So I don't know of a suitable analogy, but if you had something growing and it's sort of moving out to the outside.
Okay, thank you.
Okay, so there's this just one little step, right, where you're taking an amine group off of the cytidine, and in the dehydration step, you're making that into a oxygen. Okay, and that's the damage to the DNA. Your DNA doesn't like to have uridine residues, and so it will repair that. And it does that in a number of ways. Right? The first is that just replication will give you two different outcomes, right? It's going, during replication, this strand is unchanged. Okay, so in one daughter cell, you get the original sequence so that you at least don't get, you at least maintain that good response. The other strand, which is, sorry, I messed that up. It's this strand that is getting replaced, right? And then that is then the original sequence, whereas the U is converted to a T, and then during replication, you get a new base pair. So instead of a CG base pair, you have an AT base pair. Okay, so that's just replication. The other way that this is happening is that, so this is sort of the first one, right? The second way that this gets repaired is that you have these MSH2 and 6 proteins. They come in and recognize this UG mismatch, and they start repairing it, okay? And so if it comes in and it just repairs that U to a C, it can just sometimes do that and nothing else changes. But what else will happen is it will take sort of nearby ATs and convert those to a mismatch. Okay, and so then during replication, then you get, this will then become an AT pair, the original sequence, or this will become a CG pair. And so it's a mutated nearby AT regions. Okay, so there's several different versions of that, but that's essentially, it's repairing the original uracil base and then replacing that with replacing nearby AT pairs with random bases. Okay, now sort of the classical repair pathway is the UNG pathway. This is uracil N-glycosylase. Okay, so it's going to recognize a U in the DNA and just cut that U part of it out. The backbone stays there, There's just no base pair, or there's no base at that site, and that's it, that abasic site. Now, when it does this, right, if it replaces that abasic site with gets replaced with a C, then you just basically end up with the original sequence. However, it can also replace it with others, okay? And what we're not showing here is the replication step, that if you replace this with an A, then one daughter cell from this strand will get the original sequence, and the other daughter cell will have an AT mismatch there, or sorry, AT replacement. Okay, but it can replace that with any base, so it can give you a G or a C and change the sequence of the DNA, encoding the B cell receptor. Okay, so you have the original sequence, and then AID will make the mismatch. You can have proliferation, and doing so, you make one daughter cell with a changed sequence. You can have this sort of canonical repair pathway where UNG cuts out the base, and then Rev1 will come in and replace that with a random base. Or you can have the MSH proteins that will fix this original mismatch, but then mutate additional AT pairs. Okay, so that's the mutation part of this. And I know I had another version of the affinity maturation, but I've sort of made my own because I think it's a little easier to understand. They both essentially show the same thing, and that's where we left off last time, was right, you mutated the B cell receptor, now you go to the light zone and you have to test it. Okay, so here's our germinal center, okay? And this is called the cyclic reentry affinity maturation model. In reality, it doesn't actually work quite like this. I mean, this is sort of an easier way to teach it. In reality, you could do all of these steps without moving around. But this does occur in the light zone. It's just easier to kind of visualize it as a multi-step process. Okay, so our B cell here has gotten a CD40 signal from T cell. And it's going to go and form the centroblast. Okay, this is the dark zone of the germinal center. This is where you're doing somatic hypermutation. You're converting C's to U's and then repairing those, and in doing so, you can get different sequences. Then they go to the light zone where they're testing for, did that make the B cell better able to see antigen? And so they stop proliferating and they move into the light zone. In the light zone, they're going to interact with two cells. First, they're going to try and recognize antigen on the follicular dendritic cells. And in doing so, they can steal the antigen and therefore have something to present to a T cell. And then they're going to interact with the follicular helper cells. These are the cells that will, if they recognize antigen, will tell the B cell, go back into the dark zone and start dividing and mutating again. Now, if it doesn't, if there's no T cell help there, right, that B cell has either got nothing to show because let's say the mutation was deleterious and it had prevented the B cell from capturing antigen, well, it's got nothing to show to a T cell, and so it's going to die. Okay, so it undergoes apoptosis. So it's essentially saying there's nothing there, and there's nothing there to present to a T cell, which means the B cell wasn't able to capture antigen. Now, this antigen can also come, at least in theory, and oftentimes we sort of think of it coming from subcapsular space macrophages, but the main source is likely to be the follicular dendritic cells. Okay, so if the B cell is able to have antigen and it gets a CD40 signal from the follicular helper cells, then they go back to the dark zone and start rapidly dividing and become centroblasts again. Now this process goes on until you run out of antigen, right? If there's no more antigen to capture, and the B cells have gone on long enough, then they're going to exit from the germinal center and become long-lived plasma cells and memory B cells. The long-lived plasma cells are essentially going to move to the bone marrow where they set up shop for the rest of their life, just making antibodies. Okay, are there any questions on the affinity maturation model?
Dr. Blattman,
Yes.
for the previous lecture, in lecture 12, the steps include a little bit more detail.
Yeah, that's why I simplified it, because these are just the important ones.
Okay, so just focus on the ones that you're going over here.
Yeah.
Don't refer to the previous lecture.
Yeah, you can, I mean, it shows the same thing, but I think it includes more things that just make it more difficult to understand.
Okay, thank you.
Okay, so that's a somatic hypermutation and affinity maturation, right? In the dark zone, you mutate the receptor. In the light zone, you test if it makes it better. And the way you're testing is, can you capture antigen and present it to a follicular helper cell? If the answer is no, that B cell dies because that mutation made it worse. If the answer is yes, then it gets a CD40 signal, goes back to the dark zone and starts proliferating and mutating again. The other thing that's happening in the germinal center is isotype switching. Okay, and so remember, we have these different antibody isotypes. IgM is really good at fixing complement. It's the first one that you make. That makes a lot of sense because that helps accelerate the response to whatever antigen is there. But ultimately, to control things, you know, if you want a mucosal response, you have to have IgA. If it's just a general systemic infection, you want IgG. If it's something like a parasite, you need IgE. And so you have to change the function of the antibodies. So IgM is the first, or is the isotype that is expressed on the surface of naive B cells. And it's expressed with IgD, but IgD is, as far as we know, has no mechanism for being secreted. It's strictly a surface antibody or a surface B cell receptor. So IgM is main function is complement activation. And that is then leading to further activation of immune responses and eventually killing whatever's infecting you. Now, in this class, we don't break down the different IgG isotypes. There's actually 4 subtypes of IgG, but for this class, we're just going to focus on their main property is that they're made in the serum, and their main function is neutralizing whatever's coming in, opsonizing it so that it gets phagocytosed. If it's antigen being expressed on the surface of a cell, it will activate NK cells. And then some of the IgGs can actually still activate complement. Okay, so it's just sort of your general purpose serum antibody. There are also 2 subtypes of IgA, and we're not going to distinguish between those in this course. Their main function is microbial neutralization in mucosal surfaces. Okay, and the main job of IgA is to prevent more pathogen from getting in. Okay, its job is to push everything back to limit how much more infection you can get. And so then IgE is the epithelial antibody, right? And it's going to bind Fc receptors on mast cells and basophils and essentially allow those granulocytes to attack large things like parasites. So they're using this as a surrogate receptor. Okay, we went over that in the last section of the course. The question is, how do you switch? How do you go from making IgM to making IgG? Well, remember, these are different, what's the word I'm looking for? Sort of compartmentalized gene segments, okay? They're encoded by the consonant region genes. So C mu is IgM, C delta is IgD. Those are the first ones that are expressed. But in order to express the other ones, you have to cut out this DNA and move the rearranged VDJ segments closer to the other ones, okay? And so if you want to switch to IgG, which is the C gamma subunits, you see 1, 2A, 2B, and 3, those are 4 subtypes, then you have to actually move the DNA. This is a lot like somatic recombination where you're making DNA damage, and in the repair process, you're going to bring those segments closer to each other. So it looks very much the same in mouse and humans. Humans have a little bit more complicated setup, But the idea is that you just have to rearrange the DNA again. And this is controlled by switch regions. Remember in somatic recombination, we had the RSSs, right? The recombination signal sequences. The switch regions are sort of similar to that. These are telling the repair enzymes, yes, you can recombine this DNA, okay? And they're just upstream of each of the different constant region gene segments. So here's switch region mu. Well, it's not entirely clear why you always need to, you wouldn't need to switch to mu, but it needs to recognize another switch region, either for IgG, IgA, or IgE. Okay, so it's sort of like the RSSs where these switch regions are going to pair up. And what happens is that during transcription, you get transcripts from these S regions, and these are called S transcripts or switch region transcripts. And they're short complementary mRNA or RNA, I guess it's not really a messenger, that is going to hold open those switch regions. So you can see here, as you're transcribing from these, you make S transcript RNA that then is going to hold that region open, okay? And these are called recombination loops or R loops. So S transcripts hold open the DNA to form R loops, okay? Now this holds the DNA open and allows AID to come in and make many C to U transitions, okay? So AID is the enzyme that is in somatic hypermutation is just changing C to U. You get some, and then you see changes in the DNA sequence. Here, AID is going to come in, and it's going to make lots of changes. It's going to take all of these C's and make them U's, okay? And then the same enzyme, UNG, uracil N-glycosylase, will come in and cut out all of those bases, all of those uridines. And so what you end up with is a pretty damaged piece of DNA. Okay, and so sort of the difference here between somatic recombination and isotype switching, it's the same enzymes, but the difference is the R loops are holding this open so that AID has a much easier time of making these mutations. Okay, now the question, okay, now you've got pretty damaged switch region, okay? And the idea is that the repair enzymes are going to come in and recognize this. And instead of this being Rev1, which would come in and just put a random base there, there is a enzyme called APE1. Okay, so the repair is a little different. The mutations are the same, but the repair is different. And so what APE1 does is it's going to come into this switch region and recognize all of these A basic sites. You've cut out all of the U's that AID created, and then UNG cuts those out, and now APE1 is going to come in and recognize these multiple A basic sites, and it's going to start cutting the DNA, okay? And it's actually doing this both at the IgM region, but also in whatever isotype that that B cell has gotten a signal to switch to. Okay, and we'll talk about what the signals are for that in just a second. Okay, so that essentially what AID is doing is coming in and just cutting the DNA. But then the repair process is very much the same, right? APE1 has come here and it's cut the DNA multiple times here, but in reality, these two are paired next to each other. And so the Ku7080 proteins, which in somatic recombination are just binding to free DNA ends, doing the same thing here. They're just holding those free DNA ends so that you don't develop into a cancer or something. And then those get paired together and DNA ligase just joins them back up. So instead of just rejoining this, it's going to join this to over here and joining this to over here. And so it's basically cutting out that middle part so that now you have, you've brought the VDJ closer to the other constant regions. Okay, so the Ku7080 proteins are just sort of general DNA repair enzymes. They're just going to bind to the broken ends of DNA, and they're just holding it together. And this should look very similar or very familiar, because that's what they do in somatic recombination. Isotype switching is just a different version of that, where it's bringing the S regions together, and it's repairing the DNA damage by joining those instead of just fixing all of the A basic sites. So isotype switching uses some of the enzymes from somatic hypermutation, right, to damage the DNA. It uses AID and it uses UNG. AID makes the Cs to use, UNG cuts out all of those things. But the difference is, instead of repair with another base, you get APE1 coming in and recognizing all these and just cuts it, okay? This is because there's more damage to it. But it also uses some of the enzymes from somatic recombination. And I can tell you on the next exam, I'm going to put up those three things: somatic recombination, somatic hypermutation, and isotype switching, and ask you which enzymes are involved in each. Okay, so I'm telling you that right now, that's going to be on the exam. But the difference is, during isotype switching, you're using APE1 instead of the RAG proteins and Artemis and TDT. Those aren't expressed here. So instead, you're just repairing this by joining those switch regions together. Okay. Is there any questions on isotype switching?
I have a quick question.
Yes.
Can you describe the recombination loops a little more? Like, how are those formed exactly?
Okay, so what's happening is, as this B cell is expressing IgM, okay? It's actually IgM and IgD. During that process, you're actually getting some antisense transcripts from the switch region. That's here in this bluish purple color. And what that does is it just holds that region open by binding to the DNA, forming a DNA RNA hybrid that holds that DNA open. And so on this side, we don't show it, but they're basically, all these bases are just unpaired. So if we were doing this correctly, we would show all these little half lines here. Now that's what holds that switch region open, is that the S transcript. And that's essentially, that structure is called the R loop. Okay, so now that R loop is just an open piece of DNA, of single-stranded DNA, which AID loves as a substrate. AID loves to see single-stranded DNA so that come in and can start changing C's to U's. Does that clarify it?
Yeah, that helps a lot. Thank you.
Okay. Now, one of the signals that we haven't really talked about is how does it know which S transcripts to open up? You know? And that signal comes from which cytokines are being produced by the follicular helper CD4 cells. If it's a Th1 response, which we'll talk about in the next set of lectures, you get IgG S transcripts. So you're holding open the specific ones that the T cell is saying, this is the kind of response you're going to make. If it's making IL-4, then it's going to hold open IgE. If it's making other proteins like TGF-beta or IL-17, it's going to hold open the IgA. So the B cell is getting a signal from the follicular helper cells to say, this is the kind of pathogen we think it is, based on the cytokines they're making. And that then will help those B cells hold open the right the right switch region by making the right S transcripts.
Dr.
Okay.
Blattman,
Yes.
so when we're doing the somatic hypermutation, they're already just held open. They're not, we don't need like the S transcript to hold that open.
Right, and that's why you get far fewer C to U transitions in that process, because there's nothing holding it open, really.
Gotcha,
So,
okay.
So AID can come in, it's openly, it's transcribed, so it's open, ish. But the S transcripts really make this just hold it open, and so AID can come in and make a lot of mutations. Okay, so yes.
So somatic hypermutation is referring to the antigen binding region, and isotype switching is referring to the constant region?
Correct.
Okay, thanks.
Okay, so somatic recombination is, if we keep these separate, right, that's in developing B cells. We're going to have rejoining VD and J segments, okay, and that's, you can change the length of it by TDT. The RAG proteins are the cutters, Artemis is a cutter, and you have some of the same enzyme, and that's controlled by the RSSs, right? Remember the 12-23 rule. You need a 12 RSS combining with a 23 RSS. Somatic hypermutation is occurring in the centroblasts in those B cells. And you're introducing point mutations, so the length of the DNA doesn't change. That, you can imagine, that's pretty important, because if you change the length of the DNA, you're going to make a frame shift, and now it's not going to encode a protein, okay? So in that case, you're not cutting the DNA, you're just repairing it, okay? So you have these different enzymes, just a polymerase replication pathway, or the UNG and Rev pathway, okay? So that's controlled by just sort of having more open DNA structures during active transcription. Isotype switching also is going to occur in the activated centroblasts, okay? This is only, in order to get it, you have to have AID, and remember the AID is only expressed in the dark zone in the centroblasts. Okay, so some of the enzymes are the same. You have Ku7080s binding the ends, DNA ligase is doing this, but it's really the APE1 protein that is going to cut the DNA, and then you repair that. And the way that it knows where to do this is that you have the R loops forming because of the S transcripts binding to DNA and holding it open. Okay, so they each have different sets of enzymes, and please make a note here that I, for some reason, left APE1 out. It should be here. But you have different enzymes, but some of the repair processes sort of overlap, okay? So it's going to make sure you understand what the job of each of those proteins are in these three processes. Okay, now as I said, the cytokines that the follicular helper cells are producing are going to direct the B cells to switch to particular isotypes, okay? And so during systemic infections, you typically have a very strong T cell response because there's lots of infection there, and this is going to tell those T cells to make interferon gamma, okay? And this is a very common cytokine, and that's going to signal through the interferon gamma receptor through proteins called the STAT proteins. These are JAK-STAT pathways, and I'm just trying to keep it simple so that you just put in that you just are focusing on the STAT proteins. And the STATs are signal transducers and activators of T cells. So interferon gamma is going to bind to the interferon gamma receptor on the B cell and signal through STAT1, which once it becomes phosphorylated is a transcription factor. So STAT1 actually dimerizes and forms this transcription factor, and that turns on the Ig gamma S transcripts. So this tells that B cell, you should switch to IgG. Okay, and so then you get systemic antibodies. And that's because you have a systemic infection, you want systemic antibodies. Now, you don't always want IgG. Sometimes you want mucosal antibodies, or you want mast cell bound antibodies. And so other cytokines can step in here. TGF beta is produced by T cells primarily in mucosal lymphoid tissues. So in the gut associated lymphoid tissue or the bronchial associated lymphoid tissue, you get a lot of TGF beta. And this is telling the cell this is a mucosal infection, so we need IgA. And so TGF-beta will signal through the TGF-beta receptor through proteins that are very close to STAT1 proteins. They're actually called the SMAD proteins. These are also transcription factors. When they get phosphorylated, they dimerize, and that turns on Ig alpha S transcripts. So it tells that B cell, TGF-beta is binding to the TGF-beta receptor on a B cell, signaling through SMAD proteins to transcribe Ig alpha transcripts, which tells the B cell you should switch to IgA. And so because the TGF-beta is coming from mucosal sources, you get mucosal antibodies. Now, IL-4 is typically produced in response to extracellular pathogens, so parasitic infections or very large multicellular organisms, and this is infections at epithelial surfaces. Well, for those kinds of pathogens, you need IgE, so it can bind to Fc receptors on mast cells and basophils, so that those cells can degranulate and start killing that parasite. So IL-4, produced by the follicular helper cells, is then going to signal through the IL-4 receptor on the B cell and signal through different STAT proteins, so STAT3 and 6, and that then becomes a transcription factor turning on IgE transcripts. If you have IgE, you're going to hold up in the IgE switch region, and so you get preferential switching to IgE. That means that now that B cell is going to make IgE isotype antibodies. Those can bind to mast cells and enhance their ability to control infections at epithelial surfaces. So it's really the context, right? If you want mucosal antibodies, you get TGF-beta. That tells to turn to IgA. If it's a very large multicellular pathogen, you get IL-4, and that turns on isotype switching to IgE. If it's systemic, a very strong response, then you get interferon gamma, and that turns on the Ig gamma S transcripts, so the B cell switches to IgG. Is that clear for everybody? So any questions on the control of isotype switching? Okay. So the last thing that's happening, we've covered somatic hypermutation, we've covered affinity maturation, and isotype switching, which is also occurring in the centroblast. But now you're sort of talking about what at the end of this response, the thing that we want is a long-lived antibody response in memory B cells, okay? And so if we go back to our affinity maturation stage, once there's no more antigen, those B cells are going to start exiting from the germinal center. Now the difference here is that this apoptosis is happening early in response. As these cells have proliferated and differentiated, they don't no longer need that T cell signal to survive. And so even if they run out of antigen, they don't just die, they exit, okay? And they become the long-lived plasma cells and memory B cells. So let's talk about these two cells. Plasma cells have a huge cytoplasm. That's what they were named for, okay? And they're essentially antibody-producing factories. Some estimates are that a single cell can produce many micrograms of DNA per day, which is a ton of protein, okay? And those plasma cells, when they exit from the germinal center, will travel to the bone marrow and occupy a distinct part of the bone marrow, or a niche, where they just sit for the rest of their lives and pump out antibodies, okay? And I think the upper estimates are that you're making up to 1 milligram of antibody per cell per day, which if you've ever done any protein chemistry, a milligram of antibody is a lot of protein, okay? So they require this big cytoplasm because they're just an antibody factory now. And so if you have thousands of plasma cells per infection, you actually get grams of antibody per day. But those antibodies are proteins, right? They're floating around in your serum, and they have relatively short half-lives on the order of days to weeks. So they continue to need to be replenished, and so That's the job of the plasma cells. They're long-lived and they sit in the bone marrow and pump out antibodies. The plasma cells have longer half-lives, and there are different estimates, and it can vary in the type of infection, but you're talking about years. Plasma cells are long-lived, and so they survive for years, but eventually they die off. And one of the things that replenishes them are the memory B cells, and we'll talk about those here. So memory B cells have two jobs, right? Plasma cells have one job. They just sit and produce antibody. But the memory B cells have two different jobs. Here's our germinal center, and we get our memory B cells and our plasma cells. Now, the first job, as implied by their name, is a memory B cell. If you get infected with that same pathogen, those cells are able to respond second time much faster. They're already higher affinity, so it's a better antibody response. So you get infected a second time, your B cells are ready to go, and are able to control the infection faster. The second job they have, which is far less well understood, is that they will replace these plasma cells that die off. And the signals for that are not known. We don't know what tells the memory B cell, oh, there's a dead plasma cell, you got to go replace it. But we know that it does happen. So we don't know what the signals are, but the second job is to replace plasma cells that eventually die off. Now, the memory B cells are circulating just like the naive B cells were. They're going from the blood into the lymph node and looking for antigen. So why are they better? Well, they've already undergone somatic hypermutation and affinity maturation. So their B cell receptors are already better, okay? And so they can acquire directly. They've also gotten signals that, okay, you've already made a response, so next time, just go ahead. You don't need to wait for T cells. You can just go ahead and start this whole process. So they don't have to wait around for macrophages or follicular dendritic cells. They're just going straight into a response. And that's why, although you also have floating antibody, you also make, in a secondary infection, you make a much faster B cell response. Okay, so when we're talking about the memory B cell response, they don't require all of these other signals. They don't require complement receptors. They just have to see antigen. Okay, so, and they can do this if they're floating around and they see antigen, they can just automatically take it up, present it to a T cell. This T cell says, yep, you're good to go, and it forms the dark zone. Right? It's going to form the dark zone of the follicles. I don't know why they drew it outside here. But then from that, you're going to get more plasma cells and more memory B cells. And it just is a much more rapid response. Anytime you think of an infection, you're really talking about a race. How fast can the immune system go versus how fast did the infection go? Anytime you give the immune system a head start, you're better able to control the infection. So this whole process is essentially an accelerated version of the first response. And it's accelerated because now these memory B cells are better able to grab antigen. They present it directly to T cells because you also have memory T cells. And then you get this accelerated formation of germinal centers and production of antibodies. Okay, so I'm not gonna, I realize there's a lot of material here today, and so we've sort of gone over it, but I think it's important to understand how this looks, right? And if we just look at plasma cell IgG, let's say we got an infection here in the dotted line, okay? As that infection is being controlled, you have isotype switching to different isotypes, including IgG. So if I were to draw on here IgM, it would look like this, right? Because that's the first one. But then you get isotype switching to IgG. As that infection is controlled, then that sort of dies off. You get some steady state levels, right? That can last for your entire life. This is sort of this could be a huge break here between your first and second infections. But now you get a second exposure to antigen, and there's a much bigger IgG response. IgM would look like this. So in the first one, you had mostly IgM. The second one, it's going to be mostly IgG. Okay, and the IgG is the one that you want for that that infection. Now we'll cover this again when we go through the dynamics of the immune response at the end of this portion at this end of the section of the course, where we'll revisit what's the timing of all of these things. But if you think about it, your first source of IgM are the primary foci, right? This is T independent. They didn't get a CD40 signal, so you form plasmablasts in the primary foci. Then the long-lived IgG response, in this case, is coming from plasma cells. Those are the cells in the bone marrow. And so you can produce, depending on what type of infection, it was either IgG or IgA or IgE, excuse me. And then the memory B cells are just circulating, looking for the next infection. So when you get a boost or a secondary infection, you're stimulating those memory B cells, and that's why this response is much bigger. You're boosting the antibody response, and that's where the term boosting comes from. We went from this, boosted up to here. Okay, and that's the whole goal of booster vaccines. So you have more antibody, or you have antibody to that pathogen that will help control it, but then the faster response during the secondary infection speeds up this thing, and so it rapidly activates all of these different pathways. Okay, so we're actually going to end a little bit early today, and I'm happy to take questions because I know I don't want to add more information for you to sort of be able to understand. This is the skeleton bones of the B cell response. And next time we'll start covering the T cell response. So I'll take any questions that you have on the B cell activation.
Dr. Blotman? So in lymphomas, is it in somatic hypermutation, those central blasts, are those causing the lymphoma, or is it the actual lymphatic tissue?
It's
in
most
the lymph node?
it's either the somatic recombination or isotype switching mechanisms. Somatic hypermutation is just making base pair substitutions, so it doesn't really affect, or it's not likely to result in big changes in the DNA that result in activation of oncogenes. But in somatic recombination and in isotype switching, you're actually cutting the DNA. And that's where you can run into problems. And so most myelomas or lymphoma, B cell lymphomas are sort of result from that process.
In myelomas, are the plasma cells are differentiated, so they aren't undergoing that somatic recombination?
Well, in all of the lymphomas, they stop undergoing process that got them there.
Okay.
So the difference between a B cell lymphoma and a myeloma and some of the other ones are just at what stage was that cell when it formed a tumor cell or cancer cell? It's just what point was it already activated? Was this from a naive cell? Was it from a activated B cell? Was it from later in the response? That's just the differentiation of it. Okay, before everybody exits, there is a bonus question. The bonus question is, there are patients that don't have CD40. And so they have a particular syndrome. And the question is, what is the expected phenotype of a patient that doesn't have CD40? What isotypes will it have, and how does this impact that patient's ability to respond to infections? So when you're answering this, think about what CD40 does. What does it tell the B cell to do? And that question is not up on Canvas yet, but it will be soon.
I had a quick question.
Yes.
I forget, does CD40 refer to something presented by a maturing or a B cell undergoing affinity maturation to a T cell, or is it a receptor on the T cell?
So CD40 ligand is a receptor on the T cell, and CD40 is the protein on the B cell that's being recognized.
Okay, okay, thanks.
So one of the questions was, is this what the SARS-CoV-2 antibody tests rely on? They're just looking for antibody that's being produced by plasma cells. Okay, well, if there's no other questions, we'll see you on Thursday.